Hey, hello guys and welcome back to Excelsior Academy. So as far we were studying about the nucleus, right? So in the previous lecture, we have talked about how the proteins are getting transported from nucleus to cytoplasm and vice versa. And similarly, how RNAs are also getting transported, right? Now, we are going to talk about the regulation of these protein inputs, like how it is well regulated. Though we have discussed this already in one of our lecture, where I have mentioned how NF kappa B, okay, how NF kappa B is present in the cytoplasm. So what kind of regulation it is there? So if you remember, I have always mentioned that NF kappa B is bound with the I kappa B, right? So this I kappa B, once it gets phosphorylated, then only it leaves the NF kappa B and its NLS signal gets exposed, which helps in the binding to the important protein, and then it goes inside the nucleus, correct? So this is what we are going to discuss in this lecture in detail. Okay, so the transport of the proteins to the nucleus, it is an important level at which the activities of nucleoproteins can be controlled. So transcription factors, for example, they are the functional only when they are present in the nucleus. So the regulation of their import or export from the nucleus is novel means of controlling gene expression. So there's a one mechanism, like how? So one mechanism says that you mask their NLS signal. Okay, now once it is not recognizable, then these proteins remains in the cytoplasm, as in the case of the NF kappa B we have discussed. Okay, so same here, NF kappa B is found as an inactive complex with inhibitory protein called I kappa B in the cytoplasm. So already mentioned, right? So here, look at the figure. This you can read yourselves later. Now, in this regulation, what we see is that NF kappa B is there and I kappa B is attached, right? It is bound to the NF kappa B. Now, what is happening here is that I kappa B is actually masking the nuclear localization signal. Okay. Now, once phosphorylation happens of I kappa B, now phosphorylation हो रहा है, इसका मतलब कोई तो kinase होगा, right? So only kinase have that activity or the property to add phosphate group. So I kappa B में आपने phosphate group add कर दिया है. Now what will happen to the I kappa B? I kappa B is going to get structurally changed, or we can say that there will be a conformational change, correct? Now, because of this conformational change, it loses its activity towards the or affinity towards the NF kappa B. Now, what happens is that your NLS is getting exposed. Okay. Now, once it is exposed, the important comes and binds and takes inside the nucleus. So, this is your one type of regulation. Okay. Able to follow, Akshay, Tanvi?
Yes, ma'am.
Yeah, good. It is simple also, and we have discussed also many times. Now coming to the next one. So here we have got another protein called FO, which is actually a yeast transcription factor. Okay. Now what do we see here? You have got the NLS, which is also having the phosphorylation. Now what is happening here? Because of the presence of phosphate group, okay, what is happening? The important protein cannot bind to it, right? It is hindering the association between the 4,4 and the important. So here we need the regulation that dephosphorylation should happen. So once it removes the phosphate group, now the NLS is easily accessible to the important protein. So when it goes and binds and brings the transcription factor into the nucleus. Is both mechanism clear to both of you? Tanvi, is it clear? Should I proceed?
Yes, ma'am.
Yeah, it's very simple also. Now, binding to I kappa B, it masks the NF kappa B in the list, thus preventing NF kappa B from being transported to the nucleus. And in the stimulated cells, I kappa B is getting phosphorylated. Now, after phosphorylation, I kappa B goes for the degradation by the ubiquitin proteolysis, okay, allowing NF kappa B to enter the nucleus and activate the targeted genes. So this is how. So guys, here what you have to do is, this is one small assignment for you, that you have to study or make short notes on the ubiquitin proteins. Okay. So the pathway is over here, but it is composed of how many, like how many protein subunits are there. Okay. जैसे कि E1, E2, E3 mention किया है. Okay. So that is understood from the figure, but what you have to understand is E1 का क्या role है, like what are their functional roles in the degradation pathway. ठीक है? So that is what you have to make sure you study about the ubiquitination pathway. Okay, now let's come back to this figure. So what do we see? We have got the ubiquitin molecule. With the help of the ATP, the E1 is getting charged. Okay. So E1, E2, E3 ये सब क्या है? ये सब आपके ubiquitin proteasome के enzymes हैं. Okay. So ubiquitin is first activated by the enzyme 1. So this activated ubiquitin is then transferred to one of the several ubiquitin conjugating enzymes like E2. So here you can see that with the help of the ATP, ubiquitin is got activated E1 में. E1 से वो E2 में आपका transfer होगा. ठीक है? Now E3 is going to associate and a substrate protein to direct the transfer of ubiquitin to a specific target. So what is happening? E3 क्या कर रहा है? E3 is your ligase. So this is making sure that this ubiquitin is taken from the E2 and transferred to this target protein. Okay. Yeah. So this is being transferred to the protein. Now what is happening? So this step is getting repeated. Okay. So many ubiquitin molecules have been added. So once it gets added, okay, these ubiquitin molecules are added. Now this act as a signal. Okay. So this signal means that this protein will be recruited towards the proteasome. So here is your proteasome. So proteasome में जाएगा. Now with the help of the ATP, all the protein molecules will be degraded into peptides. Is this much clear to you guys? ये थोड़ा important है, and you have to understand the pathway कि कैसे follow हुआ है, like which enzyme is doing what. So Akshay, would you like to try explaining the pathway?
Yes, ma'am. So first, the the E1 gets activated with the help of ATP when ATP is converted into AMP. So when ATP is converted to AMP, E1 gets the ubiquitin with it, and then it passes on to the E2. E3 is nothing but a ligase. So E3 binds with the target protein. E2 passes on the ubiquitin to the target protein, which is present in the E3. Then this cycle continues again and again, and the target protein has a lot of ubiquitin molecules with it. Then it goes to the protein. It is degraded into with the help of ATP. It is degraded into peptides.
Yes, good. Tanvi, any doubts over here?
No, ma'am.
Okay, so it's really very short and simple. It's clear to everyone, but still I would like to, remind you that please make short notes on E1, E2, E3, क्या काम करते हैं, कैसे करते हैं, ठीक है? Like what are the real names of E1? Like I'll just let you know here itself. So E1 का मतलब होता है, it is your ubiquitin activating enzyme, already mentioned, ठीक है? E2 क्या हो गया? Ubiquitin conjugating enzyme. E3 हो गया आपका ubiquitin ligase. Okay. अब even though I have mentioned it, what you have to do is just find out like how exactly with the help of ATP और ये कौन से subunit में कहाँ bind कर रहा है. For example, I can just tell you that, see, from E1, okay, from this E1 it is going to the second enzyme, right? So ये ubiquitin molecule जो है वो E1 के कौन से amino acid में bind किया हुआ है, ठीक है? And to which amino acid E2 it goes and binds. Are you getting the question over here?
Yes, ma'am.
Yeah, so that is what you have to find out. एंड यू हैव टू गेट दी आंसर. ठीक है, शॉर्ट नोट्स बनाना है. इट्स वेरी सिंपल, ऐसे कुछ ज्यादा वो नहीं है. रेस्ट ऑल आई हैव कवर्ड ओवर हियर. इट्स जस्ट दैट you have to remember like which amino acids is getting involved when ubiquitin molecule is getting transferred. So it's actually very simple. If you know the structure of like ubiquitin, तो आपको उससे guess आ जाएगा कि कौन से amino acid में वो add हो रहा है. ठीक है, yeah. So that's all.